All right, sorry for the short delay. Thanks very much to the organizers for inviting me here. Today I'm going to talk about, we're going to move to the genetics now because we've heard a lot about networks, and I think this sort of nicely feeds into what I want to talk about, which is basically this kind of new methods we have where we use networks of co-ancestry to learn something more about the genetics of coded ware, but also it's going to be a little bit more about broader step ancestry and what we have learned from these new methods over the last couple of years. To start with, I think as a geneticist, you might argue, particularly myself as a population geneticist, that the year 2015 is sort of the year zero for us because it's essentially the birth of step ancestry. You don't really have to, I don't really have to explain much about that. Obviously, these are just some plots that I think most of you will have seen before of our paper in 2015 and Haag et al., where really the term step ancestry was put forward for the first time, and where we could see if this works, right? So we could basically show in our paper here that you can see this green component here in this first published coded ware individuals that would lead link the coded ware to the Yamnaya, and then we also our colleagues Wolfgang Haag et al. Showed that basically if you can model these early coded ware individuals, something like 80% Yamnaya-like ancestry. So clearly an indicator of spread of step ancestry into Europe at that time. In 2021, I was part of a sort of a nice effort to try to sort of catalog what was present, what we knew at that time about coded ware individuals from a genomic point of view. So this is like a classical PCA plot where you can see over here would be the hunter-gatherers, up here individuals from the Yamnaya background, and then here we have coded ware individuals. And the take-home from this is really that we can individuals with step ancestry here. Just need a bit of water. Sorry. I think probably a bit too much wine yesterday at the dinner. Sorry about that. Okay, yeah. So we have in this PCA we can see the coded ware individuals carry some step ancestry, and what didn't make it into that book was a paper was released in the same year by Papach et al., where we can see there's actually quite a lot of diversity in the early coded ware individuals. So this is a PCA plot from them where you can see again up here would be Yamnaya individuals from the steppe. And then all the green dots in this kind of nice transect from Bohemia are individuals of coded ware origins. And you can see that there's actually this huge spread here, right? So we have some individuals up here very close to the steppe, and some down here that are really pretty much looking like early farmers, European farmers in terms of their ancestry. So very, very broad diversity in terms of genetics in Bohemia. And this is sort of the plot that summarizes it all there. So it is really nice transect. You can see a lot of things changing here, from the farmers to the transition to the steppe, and sort of a very dynamic place. But what you can also notice here, I think, which is I think, even though it's published in 2021, is that all of these ancestry changes we can see here in the timeline, they're still framed with respect to this kind of very, very deep ancestry components that we have established in 2015, right? So you can see it's Neolithic ancestry, Western hunter-gatherer-related ancestry, EHG, and steppe-related ancestry. So it's not really going very close to the actual sort of more proximate people that were actually there, that are associated with coded ware, with global amphora, and so on, right? And so that's kind of where we still were in the genetics up until maybe last year, the year before, when we did have all of a sudden some new methods available. Where we can sort of revisit some of these questions that are still to some extent open, right? So we basically, have questions like what are really the sort of more proximate ancestral populations of coded ware? So what were the fine scale dynamics of the expansion? How did they interact with other step-related populations? How can we answer that? Well, more data. I'm never going to say it's not good to have more data, right? So we want more data always. But the thing is, we do have a lot of data already. And so what we can do to this data that we have already is actually use sort of our newer methods that have been developed over the last couple of years to really learn new things from the already existing data. I think the thing that has changed sort of most in the last year that allowed us to do that is this thing called imputation. So I don't know if someone already, because I only arrived yesterday, presented some of related work like that, but basically what genotype imputation allows us is to take the data that we already have, which is of a reasonable quality, but it's usually low coverage, and use a sort of large reference panels of modern populations to statistically predict genotypes at positions where we either don't have any kind of sequencing coverage or at least not a lot of coverage. And what that allows us is basically to then analyze this data in a new way that is very similar to what we would do with modern genomes. So we can actually use more high-resolution methods that we couldn't actually use before. And this is just from a paper from Barbara Motta, so in Olivier Delano's group, who was working with us on that, where you could just see that if you downsample high-coverage genomes to about 1x coverage, that the position in the PCA almost doesn't change. So these predicted genotypes are as good as having a fully high-quality genome with a certain limitation, of course, in terms of minimum coverage, but nevertheless, it works really well. And so one of the methods that we can actually use now that we couldn't before is this thing called IBD sharing, which I think has for sure come up already. So we're basically trying to trace these kind of segments of genetic material that is inherited identical by descent between two individuals. In this case, something like 4 generations back, they had a shared common ancestor here. And these kind of methods are really powerful, really to trace fine-scale population structure, recent relatedness, and so on. And so this is just an example of this type of regions, what you would expect if you have a common ancestor 1 generation back. This is the kind of distribution of segment lengths, and if it's 10 generations back, it's shifted to the left. So you're getting shorter segments, but nevertheless, there's a lot of information in this kind of data for trying to understand recent relatedness among individuals. This is an example of how you can use this. So one thing you can actually do with these segments is that because there is the theoretical expectation of how many long segments, for example, you would expect a certain number of generations back. This is from Harald Ringbauer's paper from last year, where they published one of the methods, tools to actually infer these segments. And they could show, for example, that these two individuals here, both of them, I think, associated with the Afanasyevo culture, they were sharing a lot of segments, and this has to be direct, even though they are very far away from each other, right? And because of the fact that they share these very long segments with each other, means that they have to have a common ancestor only a few generations back, so that there's a clear, direct evidence for long-scale, long-distance migrations within a few generations. So that's really the information that we can gain from these methods. What we can also do, though, is we can use them also just as a general similarity metric. So this is an example from our recent paper where we had a total of 1,492 imputed individuals, and this just shows basically kind of a heat map between each individual, like how similar they are based on just summing up all these segments and asking, how similar are they? And it doesn't really matter what is what here, but you can see there's definitely like a big patterning there. So there's some individuals that are more closely related here, and then there's some, like here, they're not very closely related, some pairs. So that's the kind of information we use here. And that information can actually also be represented as a network. So each link here, each dot here, sharing between two individuals can be framed in a sort of a network scheme where you can say, okay, we have two individuals here. If they share at least this amount of segments or this amount of DNA with each other, then we'll link them in a network. And then you can plot it in a nice plot like that, and actually turns out, all these different chunks here, where these are like communities that are very closely connected in terms of recent ancestry sharing. It turns out that nicely recovers a lot of the structure that we know already about the ancient genomes from those time periods. So here, this would be all the hunter-gatherer individuals. Here, there are different Neolithic farming groups. And these would be the steppe-related groups, and even you can see some that are sort of intermediate between them here. They have sort of affinities between both hunter-gatherers and farmers, so this would indicate individuals with higher hunter-gatherer ancestry, and so on. So there's really a rich amount of information in those IBD sharing networks. So what can we do with those? What we did for this paper was that we basically just used, so if we just take a step back here, we just used these networks to actually define our populations, our groups, our clusters, whatever you want to call them. So we basically just say, let's find, using network approaches, something like it's called community detection, let's find these clusters of sort of highly interconnected individuals, and then we just use those as our units of analysis, right? So we don't use any other information, we don't use archaeology, we don't use date, whatever it is. We just use the genetics to define our populations, just to see what these kind of closely related networks, what kind of genetic ancestry they capture. And turns out that actually works really well, and often, very often, closely sort of matches the either geographical, temporal patterns of the individuals or also cultural affiliations. So this is one clear example where we got this cluster of about 9 individuals, I think, and of course this is sort of the 3 strongest links between each individual, and these are individuals associated here with the Yamnaya culture and here with the Afanasevo culture. So that clearly like recapitulates that link that we all know, of course, exists between the Yamnaya and the Afanasevo, and they have very, very strong recent ancestry links as well, and this is basically captured in this case as being one genetic cluster that we use. But we can also ask how are they related to other individuals. So this particular plot shows now for those same individuals, not the links between them within its own genetic cluster, but actually with what are sort of the strongest links with other individuals. And as you can see here, if you plot them, you get a lot of these kind of green dots here that are closely linked to these step groups. And it turns out that almost all of those here are actually from the early corded Ware cultural contexts that we have, at least in this data set, right? So that actually means that there is some very, very strong links between the corded Ware individuals here and Yamnaya. And of course, this is also not new, but it's just another way, or sort of a newer way to show that it's not only like shared ancestry with the steppe, it actually has to be quite recent, because there's these very, very strong links between them. Okay, and then sort of the last thing, and sort of the major thing that I'm going to talk for the rest of the talk now that we can do with these types of data is that we can use them in similar style to previous methods, where we just use a kind of a supervised modeling approach, where we can ask, okay, we have all these pairwise relationships, we know that individuals share more with this group than with another, and we just combine these in a particular modeling approach, where we say, how about we take all these individuals here and we model them as descendants from 3 different putative source populations. So in this case, brown here would be Anatolian farmers, yellow would be Caucasus hunter-gatherers, and this is these like Middle Dawn hunter-gatherers we published as part of this work. And again, these are all defined just on the genetics, right? So we don't do any other information. This is just individuals representing a certain type of ancestry that is reasonably homogeneous and associated with the individuals from these particular temporal or spatial contexts. So when we do this here for the individuals we have in that data set, so corded Ware, here Batlax culture from Sweden, and then Sintashta here as a kind of another example, you can see that basically what varies here is a lot of the Anatolian farmer ancestry, so we get kind of quite a lot here in some of the Estonian ones, and not so much here in Poland and Latvia. But we can also move forward then and say, well, this is quite distal models. What about more proximal models? And then here we have a lot of different putative sources, including steppe. And there you can see now that in particular those early, again, corded Ware individuals from Latvia and Poland are almost 100% steppe ancestry. And then a lot of the sort of what previously was Anatolian farmer ancestry now becomes this farmer ancestry related to global amphora, right? And it's not because we used only global amphora, we used all sorts of different farming populations as potential sources. So you can see here in some of the Swedish ones, there's a little bit of Scandinavian farmer too. But even though we consider all these different later farming, Neolithic farming groups, it is basically the global amphora that gets chosen as the kind of the source of the predominant source of farmer ancestry in those individuals. And that actually extends also here to the Sintashta. You can see they have the same thing. There's some additional sort of more Central Asian Neolithic ancestry down here, which I'm not going to get into now what that might mean. But you can see that you can actually, even at the individual level, nicely see changes in ancestry and their composition and variation. So when we plot these here, this is one way we showed that, where we can say, well, if we plot here on the x-axis for each individual in the data set, the amount of steppe-related ancestry in this model, and on the y-axis, the global amphora-related ancestry, you can see here in particular these individuals here that are with these black diamonds. These are all the individuals from corded Ware context, and And they kind of fall on this line, which means they're modeled exclusively as steppe plus global amphora, right? So this is kind of a cline of increasing global amphora ancestry. So you have some early individuals with almost none, and then sort of increases all the way up to here, right? What's also interesting, though, is when you look at the sort of other samples here, and again, the ones with the colorful plots are samples earlier than 3,000 years ago, and the rest is from later. Excuse me. You can see that then there's another sort of line that goes down here now, which basically means that you have now, when steppe ancestry decreases, global amphora ancestry also decreases. But it's actually, whenever you have a certain amount of steppe ancestry, you also have global amphora-related ancestry, right? So it does look like global amphora admixture has had a very, very large impact on sort of the formation of the early steppe ancestry, not only for the coded Ware individuals, but actually it's present in almost all the individuals that we have, wherever we have steppe ancestry, right? So there's some sort of very deep basal interaction between global amphora and steppe-related ancestry that sort of forms the base of a large part of the later gene pool of individuals that carry steppe-related ancestry in Europe. So yeah, so this is kind of the take-homes until now. So we have this recent co-ancestry linking coded Ware with Yamnaya and Afanasyevo, and then we can see that actually, yeah, there's some prominent role for admixture with global amphora in the formation of these early steppe carrying groups. Okay, so new methods. We're going to have a little bit of new data also, even though this was sort of data generated for a slightly different context. But for this whole second part, I'm going to focus on a study that we have, the preprint has come out, I think, a couple of weeks back. So this is all work by Hugh McCall in our group. And so in this particular case, the sort of the downside of the previous work that I showed you was that we had only used the shotgun sequencing data that was available until that time period, because it was the first time we used these kind of imputation methods. We didn't really know how to behave with data that was generated in different ways. But in this particular context here for the new study, I mean, there's also new reported individuals, a lot of them from Scandinavia, from different time periods that is not of our interest here. But as a sort of a side effect of that, Hugh also compiled like a massive data set of individuals with both shotgun sequencing and capture data, which is this other approach that is widely used by our colleagues, where you basically enrich for certain genetic variants only and not sequence the whole genome sort of randomly as you would do in a shotgun approach. And actually it turns out you can also impute the data like that, so but you have to just be a little bit more stringent in terms of the cutoff. So this is just an example showing you on the y-axis the quality, if you want, of the imputed genotypes, and in blue is the shotgun data, and in red is the capture data, and so this is sort of ranked by how deep a sample is sequenced. So you can see that basically the take-home is that red is always below the blue, but you can actually get reasonably high also even with the capture process. You just have to use a higher cutoff, and then you can sort of jointly analyze them with them. The reason we do that is that unfortunately the way that it is for us is that there's quite a biased representation of each data type across time and space. So you can see, not surprisingly, in Northern Europe there's a lot of shotgun data because that's where we generate a lot of data, but in other places, England, for example, Central Europe, it's mainly capture data, right? So these two things are unfortunately distributed like that, but if we want to jointly analyze them, we have to use both types of data. Okay, so what did he find? So when he do the same kind of clustering approach with this larger data set, so here's a PCA again, where we have hunter-gatherers here, farmers here, and then the steppe-related people are up here. And this is just a map showing the individuals that are highlighted here. So Hugh basically found there are 4 major steppe ancestry clusters that, again, they emerge automatically from the genetic data from these recent co-ancestry links, right? So we don't label them in any other way. So there's one that is mainly Yamnaya related, so these are up here in the PCA, also in this kind of place here on the map. There's a cluster he calls coded Ware East, which is on this cline from the Yamnaya towards sort of the bulk of the later European samples and towards the farmers. And this is the spatial distribution of those samples. There's a second one, coded Ware North, which contains a lot of the Scandinavian samples, and then the sort of later samples follow on a different kind of cline here. And then there's a Bell-Beaker cluster, which is basically from, there's a bit of overlap with the coded Ware cluster, and then it projects further towards the Neolithic farmers, and it has this more Western European distribution. Right? So again, this is just clustering of the individuals. So this is just individuals that are falling within a certain context of genetic similarity, which already is, as you can see, is very informative and useful. But what's even nicer is you can use them in the same type of way that we did before with this kind of ancestry modeling. So now Hugh has used these deep early primary step ancestry sources as source clusters for other individuals later on during the Bronze Age. And then what is plotting here is that each dot here shows the predominant step ancestry. So we can distinguish those three different step ancestry sources. And so this shows the predominant step ancestry for each individual in color. And then underneath these shapes is one of those nice maps of the distribution of those archaeological cultures during that time, right? And I think, hopefully you'll be able to appreciate how, surprisingly, amazingly overlapping this is, right? So whenever you have green dots, and it's mainly the coded Ware source genetically in terms of the step ancestry, it's in the coded Ware archaeological regions, and then beakers are here, and then even here in the sort of southeastern Europe, we have mainly, we have some Yamnaya related. So there is this amazing correlation, which I was quite surprised when I saw it still, like how well it actually fits, between the genetics and the archaeology. So what else can we learn from that? So this is sort of the last bits now, where I just want to walk through a couple of models to see some basic patterns that from his data. So here I've just basically selected all the individuals that had some cultural, archaeological culture, contextual information related to these major groups. And this is a very distal model again. So we start with steppe ancestry, so Yamnaya related Anatolian farmer and hunter-gatherers. And so you can already see that there's sort of some major differences in terms of lots of steppe in the coded Ware. Bell-Pickers has more farmer ancestry. There's some here even with full farmer ancestry. And then later here Unetice maybe back to more steppe. But that's not that interesting because that kind of goes back to what we had before already. So just framing it in terms of these deep sources. Now if we move a little bit forward and say, okay, again, which different farmer groups do we have? And again, you can see this major impact of global amphora, which is this gray component. So it's really sort of high all across, no matter which cultural context. But you can also see there is a different farming component, like a Central European farmer. So there's some Bohemian Neolithic samples that are used for this particular source that seems to be present in a lot of the beaker samples, but not so much in the other individuals, right? So already there are some differences between, ancestry between those cultures. And then the nice thing is now when you actually go back to, and this is the same data as for the map before, when you then go to the actual steppe ancestry carrying early groups, you can see this clear distinction, the coded Ware, Fatianovo, it's all like modeled from this early coded Ware northeast samples. The beakers, there's a bit of variation, but certainly mostly central bell beaker type ancestry, and then Unetice goes back again to more coded Ware like ancestry, right? So actually, quite amazing how we can distinguish just purely from the genetics, individuals from those different groups when you go into sort of more fine scale ancestry. We'll just look at coded Ware in particular. You can see there's also a bit of fine-scale structure there. So this is, here we have Fatianovo, and then we go for sort of from northeastern Europe to Central Europe. So you can see there is a bit of an increase towards Central Europe, in particular in Switzerland, of this central bell beaker ancestry, which I guess makes sense. It's not exactly the same steppe source for all of them. There's probably some interactions with beakers in that particular part, or just in general some structure among the different steppe groups between those sites. But those are the kinds of things we can then, of course, follow up with some even more local models, which is not something that I've done here. And then we can revisit that kind of transect again from Bohemia. So we have seen there seems to be a lot of changes there that are happening, even when you look at it from a very distal point of view. So this is just to show you that we recover that distal model from the farmers to the hunter to the steppe with an increase in hunter-gatherer ancestry early on in Bohemia, in the Neolithic farming ancestries. Then we have no farmer ancestry in the earliest coded Ware samples. You can see here there's very little, then there's a little bit more later on. There's this very low amount of Baltic hunter-gatherer that is always required, so that's the red part here. That also sort of nicely links with the results from Papac et al., where they were showing that there's some link of these early coded Ware samples with, I think they used a Latvian source there, so this is also like nicely recovered by this modeling. And then again, increased farmer ancestry in the bell beakers, and then a sort of a resurgence of steppe ancestry, if you want, in the Unetice. Then we move forward, again, with Central European farmers. Now, same as before, we do see, generally speaking, global amphora as the farming source. With just in the Beaker period, we do see this kind of Central European farming ancestry appear in this Bohemian transect, but it kind of disappears again after that. Yeah, and then same story as before, right? But now really nicely seen, not just across Europe, but in one particular place, right? So this is all this Bohemian transect. Early on, mainly corded ware related ancestry, then it switches to Beaker steppe source, and then back to corded ware. And actually, this is the local transect at this one site in Vlinves, I think you say that correctly. So these are all the individuals that in this analysis we could use there. So you can see here the earliest one is actually an individual with farming ancestry, although it does have this light, like light blue here a little bit, which is central Beaker, so it maybe would point to some interactions with some early steppe sources. Yeah, haven't really followed up on that very much, so have to see if that holds with different models too. But then we have in the early corded ware here, actually the earliest individual here needs even more steppe ancestry than that particular corded ware source provides. So this kind of reflects the decrease in steppe ancestry over time in the early corded ware. And then once the Beaker period starts, we see these different components appear and also more of that central Beaker component. And just as a last bit, when you go really even further into sort of more proximate models, we include some Baltic Bronze Age source. You can see that actually this individual here is modeled as sort of 30% deriving from the Baltic, and that's individual Vli 50, which is basically here on the PCA. 51 here was mentioned in Papac et al. As one sample that falls exactly on the Baltic Bronze Age cline here. That unfortunately didn't have enough coverage for us to do this kind of analysis, but it looks like 50 here. From a PCA, you wouldn't be able to tell that this is in some way related to this, right? It just falls here broadly on with the same sort of samples from maybe in Bell Beaker genetic diversity, but clearly it has some affinity to the Baltic. So we can even on an individual level show now influx from ancestry from different parts of Europe that actually on a PCA with this kind of more distal models would be very, very difficult to disentangle. And I think I managed to get through now, so this is just a recap from the previous two results, and now I think second part take-home messages is that there's really this amazing correlation in sort of this early genetic ancestry clusters with archaeological cultures in the 3rd millennium. And then, with these recent co-ancestry methods, we can really dive into the fine-scale genetic structure of these individuals, and even at the individual level can really start to disentangle very, very interesting processes. So, that's all I had. Thanks very much. I'm happy to take questions.